Other day we have completed pollination. As you know, pollination is of two types. One is self-pollination and other is cross-pollination. Self-pollination is also called autogamy. Cross-pollination is called xenogamy or allogamy. You know the meaning of both. The process also. Today we shall study a very important topic, outbreeding devices. For 5 marks, very important one. Outbreeding devices. Sometimes it is studied under the heading contrivances or adaptation for cross-pollination. But in NCERT text, it is explained or discussed as outbreeding devices. See, what is outbreeding devices? How to define? The devices are the mechanism to prevent self-pollination and to promote cross-pollination are called outbreeding devices. Means to prevent self-pollination and to promote cross-pollination, some mechanisms are there, some devices are there in nature. Majority of flowering plants produce bisexual flowers. You know, bisexual flower means it consists of both stamens, the male reproductive part, the pistil, female reproductive part. So, every chances are there for self-pollination. If the flowers are bisexual, the chances are more for self-pollination. If continuous self-pollination takes place, what happens? It leads to inbreeding depression. You have studied the disadvantages of self-pollination. If the self-pollination is continued for several generations, many weak characters are expressed. Weak means the recessive characters are expressed. Say, for example, in pea plant you take the tall character and the dwarf character. Tall is a dominant character. Dwarf is a recessive character. The recessive characters expressed. Then the yield of the progeny also very less. They cannot adjust or adapt better to the environment. The inbreeding depression also expressed. To overcome this, the nature always encourages cross pollination and try to suppress self pollination. How it is achieved, that is very important. The self pollination is suppressed in bisexual flowers by developing some mechanisms we call devices called outbreeding devices, also called contrivances for cross pollination or the adaptations of cross pollination. But anyhow, as I told you earlier, it is discussed under the heading Outbreeding Devices. Now, what are the outbreeding devices developed by plants to prevent self-pollination? One is unisexuality. Unisexuality. It means the plants produce unisexual flower. The flowers are unisexual. Means the male flower and the female flowers are different or separate. So that cross pollination is must. Self pollination is highly impossible. In unisexual flower, cross pollination is the only method. This is how the plants suppress self-pollination and promoted cross-pollination. Say for example, papaya. Have you heard about papaya? The papaya plants are dioecious. Male and female plants are separate. The male plant produces male flower, the female plant produces female flowers. In this plant, autogamy is not at all possible. Autogamy is completely prevented because the flowers are unisexual. Okay, I told you the flowers are unisexual. Some of the plants produce unisexual flowers, but male and female flowers, but both of them are born on the same plant. That condition we call monoecious. For example, in case of areca nut, coconut, in maize plant. Here, autogamy is not possible. In these plants, that is in areca nut, coconut, even in castor, the autogamy is not possible. But geitonogamy is possible. Means geitonogamy may takes place. You know, why all papaya plants do not produce fruits means this is the reason. Papaya is dioecious. The female plant only can produce fruits, not the male plant. Okay, this is one mechanism. ನಾವು ಪಪಾಯ ಗಿಡ ಬೆಳೆಸಿದಾಗ ಇದು ಗಂಡು ಗಿಡ ಕಡ್ದು ಹಾಕ್ಬಿಡು ಅಂತ ಹೇಳ್ತಾರೆ. ಅಂದರೆ ಹಣ್ಣು ಬಿಡಲ್ಲ ಅಂತ ಅರ್ಥ ಅದ್ರದ್ದು. ಓಕೆ, ನೆಕ್ಸ್ಟ್ ಡೈಕೋಗ್ಯಾಮಿ. Dichogamy. Some of the plants in nature are bisexual but prevent self-pollination. How this prevent self-pollination means the stamen and the pistil mature at different times. This is how promote cross-pollination. Dichogamy is of two types. One is protandry and other is protogyny. Protandry. Some of the flowers or the plants are protandrous. What is the meaning of protandrous flowers? As the name suggests, proto means first, andry means andrecium. Andrecium is first means andrecium mature first, then the gynoecium matures. Example, sunflower. In sunflower, both male and female stamens and pistils are there. That is in the central flowers. Here, in sunflower, andrecium mature first, release the pollen grains. At that time, gynoecium is young. It cannot receive, or the stigma is not receptive for the pollen grains. So, cross-pollination takes place. This is one device or mechanism. The other plants, like mirabilis. You know mirabilis? Mirabilis means Mirabilis jalapa, commonly called 4 o'clock plant. Why we call 4 o'clock plant means it blossom at 4. Flowers open at 4. ಮಧ್ಯಾಹ್ನ ಮಲ್ಲಿಗೆ ಹೆಸರಿನಲ್ಲಿ ಇದೆ. ಮಧ್ಯಾಹ್ನ ಮಲ್ಲಿಗೆ ಮಧ್ಯಾಹ್ನದಲ್ಲಿ ಹರಡ್ತಕ್ಕಂಥದ್ದು. ಮೈಕೇಲಿಯಾ ಸಂಪಿಗೆ. In Mirabilis and Michaelia, gynoecium mature first. At that time, andrecium is young. This condition we call protogyny. Proto means first. Gyne means gynoecium. Gynoecium mature first at that time, Androecium is young. So, cross-pollination is must. Self-pollination is not possible. This is how prevent self-pollination. Okay, we shall see other mechanism. Heterostyly. It is third type of device. Heterostyly, as the name indicates, hetero means different. Styly means styles. Styles are different. Okay. In plant like primrose, we come across two types of flowers. How means some flowers have long style. Stamens are very short. So, pollen grains cannot reach the stigma or the stigmatic surface. The other type of flowers in which the style is very short, the stamens are very long. Stamens are extrusus. Pollen grains cannot fall on the stigmatic surface of the same flower. Self-pollination is not possible because of different height in stigma and style. Heterostyly. Self-incompatibility. It is also called self-sterility. It's very important mechanism to prevent self-pollination. Some of the plants like tobacco, petunia, so these plants produce flowers. Flowers are bisexual, Solanaceae members. In these flowers, self-pollination may take place because the flowers are bisexual. But what is there? Pollen is prevented from germination. Pollen is prevented. Prevented from germination means it will not germinate on the stigma. Why it won't germinate means there is a genetic mechanism. So, self-incompatibility is a kind of genetic mechanism in which the pollen is prevented in germination. So, self-pollination is not possible because the stigma is incompatible. Self-incompatible stigma ಅದು ಹೊಂದಾಣಿಕೆ ಆಗೋದಿಲ್ಲ pollen grainಗೆ. That is why it will not germinate. So self-incompatibility is used, or this mechanism is used by plant breeders to produce new varieties of crops. Next, hercogamy. Hercogamy means there is some physical barrier between the stamens and the pistil, so that even the flowers are bisexual. Self-pollination is not possible. Say, for example, calotropis. Calotropis means ecce. In these plants, the stigma is pentangular. The stamens or the anthers are found in the groove of stigma. Here, in this plant, the pollen grains are not free. They are found in a bag-like structure called pollinia. Pollinia. A bag-like structure contains pollen grain. So, pollen grains are not free. Even stigma is very close. Pollination, self-pollination is not possible. Cross-pollination is only the way to pollinate the flowers. So all these mechanisms or devices comes under outbreeding devices. In your textbook, outbreeding devices are not discussed under these headings. Simply the information is discussed. It is better you study like this. Very important for NEET also. NEET and CET. Okay. Now, that's all about outbreeding devices. Next one is pollen pistil interaction. What is this pollen pistil interaction? Pollination. After pollination, the pollen grains are deposited on the stigma. Pollination in angiosperms is always followed by fertilization. Fertilization is followed by seed formation, the embryo formation, and it is followed by fruit formation. Okay. Now we shall study pollen-pistil interaction. What is pollen-pistil interaction? All the events taking place from deposition of pollen on stigma until the pollen tube enters into the ovule are together called pollen-pistil interaction. Okay. Now refer the diagram also. In the diagram, the pollen deposition is there and germinating pollen is also there. Pollen tube is also there. The style and stigma you can see now. Okay. Pollen-pistil interaction. As I already told you, after pollination, the pollen is deposited on the stigma. Means we find pollen grains on the stigmatic surface. Now the stigma identifies or recognizes the compatible pollen. Compatible pollen means adjust ಆಗ್ತಕ್ಕಂಥದ್ದು ಹೊಂದಾಣಿಕೆ ಆಗ್ತಕ್ಕಂಥ pollen ಅಲ್ವೇ ಅಂತ. Who identifies? The stigma recognizes or identifies the compatible pollen. If the pollen is incompatible, it rejects. Reject means it prevents the pollen from germination. That is pollen germination. What is the meaning? If the pollen is compatible, only the stigma promotes germination. Otherwise, it will reject. So this is very important one. So when the stigma at the receptive stage, it secretes some carbohydrate or a sugar. Because of that reason, it germinates. Okay. So when the pollination takes place, the pollen grains are deposited on the stigmatic surface. That there is a continuous dialogue takes place between pollen and the stigma. Continuous dialogue takes place between the pollen and the stigma. Interaction of the pollen and the stigma depends upon the chemical components of pollen and stigma. Chemical component means the stigma secrete the chemical called the sugar, carbohydrate. Then only the pollen germinate. Okay. If the pollen grain or the pollen compatible, then only it germinate on the stigma. How it germinate? The structure of pollen grain. On the pollen grain is protected by two layers, outer exine and inner intine. Exine is thick, intine is thin. On the exine, thin-walled regions are there called germ pores. Okay, you must remember. If the pollen is compatible, it germinate and produce pollen tube. How the pollen tube is produced? The inner intine comes through the germ pore in the form of an outgrowth. Actually, we call the pollen tube. This pollen tube grows through the stigma or the stigmatic tissue, and the pollen tube grows through the style. It means when pollen germinate, the pollen tube is formed, it develops or grows through the tissues of the stigma and style, ultimately reach the ovary. You know, the pollen in majority of angiosperms, when the pollen is at two cell stage, that is vegetative stage and generative stage, the pollen grain or the male gametophyte deposit on the stigma. Now, the generative cell of the pollen grain divide. It means its nucleus divide mitotically and form two male gametes. The male gametes are non-motile, SET or neat point, and it is a highly advanced character of angiosperms. This happens in some plants. That is, two-celled male gametophyte having vegetative cell and generative cell. The nucleus of the generative cell divide mitotically and produce two non-motile male gametes. But in some 40% of angiosperms, the generative cell already divided mitotically and contain two male gametes. At this time, pollination takes place. That is three-cell stage. So to make it brief, after pollination, the pollen grains may be two-celled or three-celled. Okay. Now, the next part. The male gametes are transported or migrated to the tip of the pollen tube. There is vegetative nucleus also there. You refer the diagram. The diagram showing the tip of the pollen tube with two male gametes and even vegetative nucleus. The pollen tube enters into the ovule normally through the micropyle. This is called porogamy. Then the pollen tube enters into the synergy. At the micropylar end, the synergids contain a special kind of cytoplasmic structures called filiform operators. The synergids contain or consists of filiform operators. What is the function? the function of filiform apparatus is it guide the pollen tube to enter into the embryo sac or female gametophyte or into the what we call central cell, or even we may call ovule. So very important point for neat examination. Now see the diagram showing pollen grains deposited on the stigma, germinating there. A diagram, the B diagram, pollen tubes are growing through the style. ಎಲ್ಲ ಥ್ರೆಡ್ ಲೈಕ್ ಸ್ಟ್ರಕ್ಚರ್ಸ್ ಕಾಣ್ತಾ ಇದೆ. That is the one. Next, you can see now the diagram C. Here the pollen are deposited on the stigma. Some two pollen are just germinating and the other one produce pollen tube. The pollen tube entering into the ovule through the micropyle, what we call porogamy. In the diagram D, you can see the enlarged diagram, how the pollen tube enters into the synergid. You can see synergid having filiform apparatus, cytoplasmic thread-like structures. You can see now the pollen tube, the tip containing the male gametes, even the vegetative nucleus also. The egg apparatus you can see. In the diagram D, the tip of the pollen is open or burst. Two male gametes released. Okay. All these happenings we call pollen-pistil interaction. That is, from deposition of pollen on the stigma up to entry of pollen tube into embryo sac or ovule is called pollen-pistil interaction. Okay, so that's all pollen-pistil interaction. Next topic. What happens to this? Andre pollination followed by fertilization. Next is fertilization. That's also very important for the examination. So this pollen-pistil interaction is very, very important for 5 marks. Okay. Thank you.